In this video, we're going to continue our discussion of ion channels. Now, ion channels aren't normally left open, rather they are normally shut and only open for a very brief moment to perform a specific task. And we call this gating. And there's 4 different types of channels out there that are gated. So first we have the mechanosensitive channels, where if the lipid membrane changes shape, it can open. We have a ligand-gated channel, where if you bind a ligand, you will open. Signal-gated channel, which if you bind Ca2+ or something else, you will open. So this is intracellular, and ligand is extracellular. And voltage-gated channel, which if you have a change in the charge of your membrane, you will open. And the best way to talk about this is nerves. So neurons are voltage-gated channels. So if your neuron gets neurotransmitters, this binding will allow sodium channels to open. And when sodium channels open, a bunch of sodium will flow into the neuron. And so when that happens, you have local depolarization. So let's take a look. So normally the inside of your cell is negative. So you get a signal that tells your sodium channels to open. A bunch of sodium flows in, and now your inside of your membrane is positive. When you get this local depolarization, your potassium channels open. When that happens, potassium flows out of the cell. And so now you go through what's called hyperpolarization. Your cell membrane becomes extremely negative. And then both your sodium and potassium channels will close, and you'll slowly go back to your base level. And what actually happens is that we can propagate a signal throughout the whole neuron by this mechanism. So if the signal starts here, bam, we're letting sodium and potassium in, and when we do that, our channels next to us are going to open in response. So when a sodium channel opens, neighboring sodium channels will also open because they open with this positive. Potassium opens as well to shut off the sodium channel that was next to it. So you have this very brief change in membrane potential that travels the length of your neuron until it ends here. Then you can send a signal to the next neuron, more or less propagating the whole signal throughout your body. And so that's how you can send signals through your neurons. It's just voltage-gated channels to do with sodium and potassium. Now, this voltage-gated channel, we call it the KV channel for potassium, V for voltage, so potassium voltage channel. And they're all relatively the same in that you have 220 residues of your N-terminus that's in your cytoplasm, shown here. You have 250 domains that are transmembrane and 6 helixes, shown here, and roughly 150 residues of your C-terminus. So this very much looks like the potassium channel we were looking in the last slide. Now the way that this voltage-gated channel works is that on the 4th transmembrane helix, you have 5 positive amino acids that are highly conserved. This is your voltage sensor. Now, when your sodium channel opens and a bunch of sodium flows into the cell, the inside of your membrane becomes positive. When that happens, the positive residues on this channel will get away from the positive charge inside your cell. They will move to the outside of your membrane. So these arginines, I have 4 here, but I should say 5. These arginines are pulled towards the outside. When you pull this to the outside, your pore, which is made between S5 and S6, they get pulled as well. That will enlarge this channel, allowing for potassium ions to flow out. Then you'll get hyperpolarization, where the inside becomes negative again. When the inside becomes negative, these residues, these positive residues, will want to be pulled towards the inside again, effectively closing this pore. And here we can kind of see that again from a different look. So what you need to consider is that this drawing is actually showing like a ring-like structure. So that is how it's opening. So we can't really show this this compound well on a 2D space, but just know that this is forming a pore. These are going around in a circle, even though they're flat like this. Now, when this channel opens, it closes extremely fast. And studies have shown that if you chop off the N-terminus, that you can no longer shut off this channel extremely fast. Therefore, the N-terminus has been called the inactivation ball, because you have residues here that will swing in to the pore and actually bind to the opening and block any more potassium from leaving. So when this pore opens, this ball, imagine it swinging into the pore to immediately shut it off. And it's probably not like being a ball that enters, rather these first 10 residues are incredibly hydrophobic, and there's hydrophobic residues in the channel. So what probably happens is that this ball snakes in and binds to the hydrophobic residues and forming a good plug like that. And there's other channels that work with the same idea for sodium and calcium that follow the same idea, where you have this pore and it looks the same, inactivation ball, and same mechanism. So we're just looking at potassium for our model. Now, if you look at a negative ion channel, that is something that transports an anion, such as chlorine channels, we find these in all cell types. The extracellular chlorine is high, intracellular is low, so you don't want a lot of chlorine into your intracellular membrane. And these are homodimers, so it's a protein made out of two smaller proteins are the same, homodimer. And you again have a selectivity pore, that's an anion pore here. And so the subunits are made out of transmembrane alpha helices, as shown from this picture, a bunch of alpha helices. And the anions funnel to the pore via basic amino acids. That is, you have positively charged amino acids on the surface that will bind your negative amino acids. Then, once in the pore, chlorine will interact with amide nitrogens and hydroxyls for serine and threonine to be passed down like a bucket brigade, like we talked about the potassium pore. And you have an hourglass shape with glutamic acid and any other negative pore that or negative ion that tries to interact with the pore, they will be inhibited by glue, because remember, glue has negative charge, and so those two will repel each other. But chlorine can go through. So as we see from the negative ions, although they have a different shape, the idea is the same as the potassium channel that we have talked about previously. That's it for this one. I will see you in the next video.